little more closely at how the molecule 2,3-bisphosphoglycerate, or 2,3-BPG, regulates hemoglobin's affinity for oxygen, and why this is beneficial in conditions of high altitude as well as in the context of fetal hemoglobin. Going back and thinking about our hemoglobin, we have those 4 subunits, 4 binding sites for oxygen, and oxygen binds cooperatively. With positive cooperativity, meaning that if one oxygen binds, then the other sites are kind of like bind quicker, and if one lets go, then the rest are going to let go. And the biochemical basis of this is that when you have these 2 forms, the taut form when it's deoxy and the R form when it's relaxed and oxy. And so T is for taut. And basically, there's the salt bridges that form between the subunits, between the different chains of the hemoglobin that are able to form when the oxygen is not bound. But then when the oxygen is bound, it kind of yanks those apart, and you're not going to get those salt bridge forms. You get this relaxed form that now all of these subunits are going to have a higher affinity for oxygen. And what's going to happen is then you're going to go from this transition from a low affinity state to a high affinity state as you increase the concentration of your oxygen. Because now what's going to happen is one oxygen binds, the rest follow, one lets go, the rest follow. But remember that you're continuously going to have binding and unbinding, binding and unbinding, but you kind of are going to overall reach this state where some proportion of it is bound. And so our Y, this is telling us like what proportion of the hemoglobin sites are bound. But remember that because we have this cooperative nature, it's not like you're going to see most of them have 2 bound. Instead, you're more like at the halfway point. Instead, it's more likely that you have like half of them be unbound and half of them be fully bound. So half of them would be like in that T state and unbound, and half of them would be like in the R state and bound. Now we can think about kind of shifting this curve. So if you look at the kind of just the general curve of the hemoglobin binding to oxygen, what you see is that, so on our x-axis, we have PO2. This is the partial pressure of oxygen. Basically, how much oxygen is in your lungs or in your tissues. Basically, this is just the amount of oxygen. We can see that in our lungs, we have a lot of oxygen. What this means is that the hemoglobin is all going to basically be bound to your oxygen. And so you see your Y, the proportion bound, also like your theta B, you can call it, is going to be almost 100. There's lots of oxygen around, and you're going to be in that high oxygen affinity state because of that cooperative binding, where if you bind an oxygen, you're going to quickly bind a lot more. And even if you were to let go of an oxygen, there'd be another one to bind right back. And so you'd be basically saturated. What's going to happen is that in your tissues, you're going to have a lower concentration of oxygen. And so if you imagine going from your tissues to your lungs, well, you are going to be grabbing oxygen as you get to the higher concentrations in your lungs, but that also means that you're going to be releasing the oxygen in the kind of like the difference in from when you're going this way to get back to the oxygen concentration in your tissues. And you can see that you kind of get this dramatic drop because as it starts dropping, well, now we get the cooperativity where if you drop one, now you're able to stabilize the T form. You're able to form those salt bridges that kind of, you can think of them kind of like kicking the oxygen out of the rest of them. Bottom line is you want to compare what the saturation of the hemoglobin in the lungs to the saturation in the tissues. So you can see here you have this partial pressure in your lungs and this in your tissues. And if we look at the hemoglobin saturation, we see that we're basically getting rid of about 1 to about like 6 or so. So basically we're getting rid of about like 0.4 or whatever of what we have, but we still have a lot left in our tissues. We still got a lot of oxygen that we're not kind of getting rid of. And this will come into play in a minute when we think about shifting this curve. So let's now think about what would happen if we had more or less 2,3-BPG. So 2,3-BPG, what is it, first of all? Well, 2,3-BPG is 2,3-bisphosphoglycerate, sometimes called like DG. And what it is it's basically kind of a mutated version of one of the intermediates of glycolysis. And so glycolysis, the breakdown of glucose, you have this step where you have phosphoglycerate kinase is going to take you from 1,3-bisphosphoglycerate to your 3-phosphoglycerate with the generation of an ATP. There's a side path, and this side path It's called the Rapoport Leubering Cycle or whatever. Basically, the side path, what it's going to do is you have this combined enzyme, this phosphoglycerate mutase, and what you get is you kind of have this mutase step, which takes this 1,3-bisphosphoglycerate from your glycolysis and shifts the phosphate, so you get 2,3-bisphosphoglycerates, and then it removes one of the phosphates, so you still end up with 3-phosphoglycerate, but in the process, you kind of gave up your opportunity to generate an ATP. And you have 2 of these per glucose, so it's energetically expensive, but it's important because you're generating this 2,3-bisphosphoglycerate, which can now go and regulate the affinity of hemoglobin for the oxygen. So in fact, this happens a lot in your blood cells. So about 15 to 25% of the glucose that gets converted to lactate in erythrocytes goes through this synthesis pathway, according to this website, Online Biochemistry Notes. So basically, this is happening a lot in your blood cells, which is important because in your blood cells, they're not going to be generating the CO2 as much because they don't have mitochondria, but this is a way in which they can still decrease the affinity of their hemoglobin for the oxygen so that they can offload it. So why does 2,3-BPG lower the affinity of the hemoglobin for the oxygen? Well, what it does is it binds in the central cavity and the central channel of the hemoglobin in the top form. So in the T form that is offloading the oxygen. If you're stabilizing the top form, you're basically it's going to be binding in between the beta subunits. It's going to be stabilizing it so that you're going to kind of have more favorable in this T state. And in order to kind of get back to the R state, you've got to kick that 2,3-BPG out. So the tighter the 2,3-BPG is bound, the better that's going to be. And that'll come into play later. But the 2,3-BPG binding itself is going to make it harder for the oxygen to bind. What this means is that our binding curve is going to be shifted to the right. We're going to have a lower affinity for the oxygen in the presence of increasing amounts of 2,3-BPG. So you can see if you don't have any 2,3-BPG, well, here your curve is basically pretty much hyperbolic. You don't even see that sigmoid really. But if you have 5 millimolar of BPG, this blue line, you see that we have the sigmoid, this nice cooperativity scene. And then if we have 8 millimolar, so more BPG, well, now we're going to shift right. And so a shift in the right of our curve is going to be a lower affinity. And so we have a lower affinity for the oxygen in the presence of the 2,3-BPG because that BPG is binding in that sort of central channel and it's stabilizing that T state. And remember, if you're stabilizing the T state, you're stabilizing the form that has released the oxygen rather than the form that can bind the oxygen. So you can imagine that if 2,3-BPG is bound, it makes it harder for the oxygen to bind. And if the oxygen can't bind, well, you want to think about things like rebinding. So remember, it's this dynamic equilibrium, binding, unbinding, binding, unbinding, binding, unbinding. And what's going to happen is that if you have the 2,3-BPG kind of sneak in, when the oxygen gets offloaded, well, now it's going to make it harder for the oxygen to bind back. And so in this way, you're going to prevent it from rebinding and therefore be better at delivering the oxygen if you have 2,3-BPG around. This comes into play because the 2,3-BPG is going to help it make it to your tissues so they can give up the oxygen. But it's also going to come into play because it has effects with altitude. So if we think about the oxygen level, so before we kind of talked about how if we went from the PO2 in your lungs at sea level to your tissues, you're basically going to give up about 38% of the oxygen. So you're going to be delivering about 38% of the oxygen. So remember that when we go to the right, that's the stuff we're kind of picking up. And if we go to the left, that's the stuff we're kind of dropping off. But remember, it's this dynamic equilibrium. And so what's going to happen is that the kind of the higher affinity you have, the more you're going to be picking up or the less you're going to be dropping. And the lower affinity you have, the less you're going to be picking up or the more you're going to be dropping. And then depending on the concentration of oxygen, well, that's going to kind of determine how much oxygen is around to pick back up. If you have 2,3-BPG around, it's going to make it harder to pick back up the oxygen if you were to drop it. And so in this way, you can think about 2,3-BPG kind of helping you offload the oxygen. So if we think about from our lungs to in our tissues at sea level, that's about 38% that we're getting rid of. But now let's imagine that instead of starting at sea level, we start at a high altitude. So at 4,500 meters, well, now you can see that we're starting at a lower level of oxygen. So our oxygen level, remember, that's the x-axis, and then our fraction of the hemoglobin bound, that's our y-axis. And so if we look here, we can see that we're at a lower concentration of oxygen, and we have correspondingly, we have a lower concentration of our hemoglobin bound. So we're starting at a lower point, but we're still ending at the same point. Our oxygen in our tissues, we're ending at like the same point in both cases, but we're starting lower if we're at a high altitude. So we're only going to be dropping off about 30% as opposed to 38%. That's a lot of oxygen that that these people are kind of that you'd be missing out on. One way that you could increase the amount of oxygen that gets offloaded is actually by decreasing the affinity of the hemoglobin for the oxygen. And you can do this by making more 2,3-BPG. So in fact, people that live at high altitudes, their bodies are typically going to produce more 2,3-BPG. So if you think this green line represents the 8 millimolar of the BPG that's produced in people at high altitudes. If we look at the concentration of oxygen in their lungs at this point, so note that because we have the 2,3-BPG there, we are going to have a decreased affinity. And so we're going to be decreasing our affinity a little at this point. But if we can compare these two lines, keep in mind how small this kind of difference is at this point, because we're still kind of nearing that saturation point. So we have these smaller changes between these lines. We're not at that steep slope part yet. So remember, then we're going to basically have the steep slope because of that cooperativity and stuff. If we have more 2,3-BPG, we're shifting our curve to the right. When we're shifting our curve to the right, yes, we make this lower a little, but now if we compare the difference here, we can see that this is a much bigger difference between if we had a lower concentration of 2,3-BPG and a higher concentration of 2,3-BPG. This difference is going to be bigger than this difference, which means that we are going to be offloading a total of 37%, which is going to be similar to the 38% that we would have offloaded if we were at sea level and didn't have the issue with if we had like 5 millimolar BPG. So if we have the more BPG, we're going to offload a little more. We'd still offload more from going from our lungs to our tissues than we would if we had started with less oxygen, but it's not that big of a difference up here in the amount that's going to be bound if you have that 2,3-BPG, if you have that lower affinity, but it is a bigger difference here. And in this way, increasing the amount of 2,3-BPG is able to increase the amount of oxygen that gets delivered to the tissues. Once the oxygen's kicked out, it's going to prevent the hemoglobin from picking it back up, because that wouldn't be very helpful if it just kind of like, yeah, it released it, but then it grabbed it right back up. So 2,3-BPG is going to help with this. Another thing that with 2,3-BPG is that it's going to kind of not affect fetal hemoglobin very much. So fetal hemoglobin, so our main adult hemoglobin is made up of 2 alpha and 2 beta subunits. But fetal hemoglobin is made up of 2 alpha and 2 gamma subunits. Right, so you have gamma instead of beta. So this is just showing you like the synthesis, the different hemoglobin subunits that are produced as a function of like time and stuff. And so you can see that initially the fetus it makes the alpha, like the whole, all of its hemoglobin, all hemoglobin almost, except for in the very initials, have this alpha on through adulthood. But you kind of switch out having a gamma for having a beta. And why this matters is because remember I said that beta was where you were kind of getting the binding of the 2,3-BPG was between the beta subunits. So if we go back and we look at the structure of 2,3-BPG, we see that it has these phosphate groups and this carboxylate group. So it's a very negatively charged molecule. If we think about the binding side of this, well, if we want something that's going to bind tightly to a negative thing, we want a positive thing. Where can we find positive things in amino acids? Well, we can find the basic amino acid side chains. So we have our histidine, our lysine, and our arginine. And in fact, if we look at the 2,3-BPG binding site of adult hemoglobin, we see that we have histidines, we have lysines, lots of positive charge that can come and bind to this 2,3-BPG, forming these salt bridge interactions, stabilizing that T form. Because remember that the T form is the form that doesn't bond, has a low affinity for oxygen, it's not bound to oxygen. If you have an oxygen bound, what's going to kind of knock this thing out. So basically cause the one monomer to, if the oxygen binds to one monomer, it kind of is then going to change shape and yank the thing next to it, and that's all going to change shape. And so all those hemoglobin subunits are going to transition to that R state, and 2,3-BPG can't fit in that R state. A channel is not there in that R state. You don't have that place where the 2,3-BPG can bind. But then whether or not 2,3-BPG combines depends on whether there's actually like a favorable site there. And so if we look at the fetal hemoglobin, so because they have the gamma chains, what's going to happen here is that they're going to have, yes, they have that lysine, but now instead of this, now instead of a histidine here, they have a serine. And so with serine, it's just got that OH group. And so you don't have that group that can actually form that favorable interaction. And you can see that some of like these histidines that would normally be forming interactions with 2,3-BPG are kind of like in the wrong position. As a result, the fetal hemoglobin is going to have a lower affinity for the 2,3-BPG. If it has a lower affinity for the thing that lowers the affinity for oxygen, well, that means that you're going to have a higher affinity for oxygen. If we're considering the fetal versus the adult in the presence of the 2,3-BPG. So let's do that. Let's compare. Okay, so if we look at them without 2,3-BPG, they're not that different. And remember, we don't have as much of that kind of cooperativity. So the ones we want to compare are kind of this HbF plus PBG, so HbF for the fetal hemoglobin and HbA for the adult hemoglobin. What we can see is that the curve of the hemoglobin for the fetal hemoglobin is going to be shifted left. Remember, we shift left, that means we have a higher affinity. It takes us less oxygen in order to kind of like fully be saturated. Often we talk about kind of like less to get to this kind of like halfway point. As 50, you have a shift left, kind of like if we were talking about a KD. Lower shift left means that you have a higher affinity. It takes less of you to basically be bound the same amount. So if we compare, like how much do we need to be at this halfway point with the fetal versus the adult, we have less needed for the fetal hemoglobin because we have a higher affinity. So basically, there doesn't need to be as much oxygen around in order for us to be found with oxygen bound. Because remember, oxygen can bind and unbind and unbind and unbind, and the more there, when it drops it needs to find a new one. The more oxygen there is around to pick one back up, the more likely it is to be bound. But basically, then you have to compare how likely are you then to drop it again, or and that sort of thing. And so the higher affinity you have, the less of the oxygen that needs to be around in order for you to be bound. So we can see that we have the shift left. We have more of the fetal hemoglobin has a higher affinity. Now, why does this matter? Well, this matters because that fetal hemoglobin is not going to be seeing those concentrations of oxygen that you saw in your other tissues. Instead, the fetal hemoglobin is going to be seeing a much lower concentration of oxygen. And so if the fetal hemoglobin didn't have the kind of a higher affinity, it wouldn't be able to grab up the hemoglobin, enough of the hemoglobin. If the fetal hemoglobin didn't have the higher affinity for the oxygen, it wouldn't be able to get enough of the oxygen. Because it's not starting out at those nice high concentrations of oxygen that you would see in the adult tissues, where if you lower it a little, it's okay. Instead, it's going to be lowered too much. And so if we had these lower concentrations of oxygen that the fetus is going to see, well, that means that basically it's not going to be picking up enough oxygen. It's not going to be delivering enough oxygen. And so we need to shift the curve to the left in order for it still to be kind of grabbing as much oxygen as it can from the mother's blood. And it's okay that it has a higher affinity because the concentration of oxygen is going to be lower. If you had a higher affinity, but the concentration of oxygen was unchanged, well, then it would make it hard to deliver oxygen. But if you have a concentration of oxygen that's lower, and now you're saying you have a lower affinity, that's okay. What this means is that you're going to basically be in that range where you're able to both grab it from the blood and offload it. And so fetal hemoglobin is going to basically not be affected by the 2,3-BPG very much. Well, I mean, it's still a bit. So if you compare the with and without, so we have the adult without BPG and the adult with, you see this like massive shift. The fetal without and the fetal with, not as much of a shift, but you still do see a shift. You still can bind the 2,3-BPG, but it's just not nearly as dramatic. And there are also things with like the pH that can be affected by the 2,3-BPG that I'm not going to get into. But bottom line, the fetal hemoglobin isn't affected as much. It doesn't have as higher, it has that mutation that makes it so that it's not going to bind to the 2,3-BPG as much. You're not going to get that shift in your curve quite as much. So you're going to have this higher affinity because you're not going to have that affinity lowered by the 2,3-BPG. And this higher affinity is going to be good because you're not going to see as much oxygen. And so you're going to be in that range where you can load and offload at a lower concentration of oxygen, at concentrations where if you didn't have the higher affinity, you would have just released it all already. So if you think about the oxygen levels that the fetus is encountering, we have our fetal arterial blood here and our fetal venous blood here. Not that big of a difference, especially when you compare the kind of your lungs to your adult venous blood. There you have this huge shift. Whereas here you only have this small shift in the oxygen concentrations. So we need to shift our curve accordingly to have it so that the steep region of the curve is in the area of the concentrations at which we're actually going to be encountering, or the fetus is actually going to be encountering. And so if we compare the difference that we would have if our fetal hemoglobin at the arterial blood versus the venous blood, we see that we get this big shift, this big offloading. But if we look at the concentrations of the adult hemoglobin, if we look at like what it would do at these oxygen concentrations, well, these oxygen concentrations here, it's only like going to be 0.25 or whatever, like only a quarter of it is actually going to be bound to the oxygen, as opposed to if we look at our fetal hemoglobin, here we're up to like half of it is bound in this arterial blood. And then if you look down at your venous blood, you've released it. And so if you looked at the adult, though, you would only be releasing basically like a quarter of the potential hemoglobin sites that were available. So by shifting the curve to the left, we're shifting the steep part of the curve into that region that is actually going to be encountered in the fetal tissue. And in this way, the fetus is able to both grab onto oxygen and release it, even though there's not that much oxygen available. So we need to have that higher affinity to begin with and shift the curve region into the kind of region that's actually going to be applicable. And although we are kind of raising the affinity, you have to remember too that there's less oxygen around, and so you're still in this zone where you have about like 25% or whatever of the oxygen sites filled. So you're going to be in this range where the hemoglobin is able to kind of like bind and unbind the oxygen, and therefore it's able to deliver it throughout the fetus as needed. Even though there's a low amount of oxygen around, it has enough affinity to pick it up, but also there's not enough of it so that if it drops it, it would quickly pick it back up. And therefore you're able to kind of have the fetus get all the oxygen it needs, despite it kind of like already being used up by the mom. The mom actually also, I read that they like upregulate the amount of 2,3-BPG that they make so that they're actually offloading more, so that the fetus has a little bit higher to begin with, but it's still in this really low range. And if you have the lower affinity hemoglobin, you wouldn't be able to pick up enough. And so you want to make sure that you can pick up enough in the fetal hemoglobin. So you want to shift your curve less. But you can imagine that we've been talking about how that 2,3-BPG affecting the hemoglobin was important for the adult hemoglobin in order to offload the oxygen to your tissues. And so the adult hemoglobin basically comes into play at birth. A little after birth, you start making the beta chain and not making the gamma chain. So the gamma is the one that the fetal hemoglobin has instead of the beta. And so they kind of swap roles after several weeks after birth. And so if babies have mutations in like sickle cell anemia or something that's affecting the beta globin, they might not even show up for several weeks because the beta globin doesn't come into play yet. But there are actually techniques that are like treatments for sickle cell and things that actually do increase the expression of the fetal hemoglobin in order to help compensate. So the main things about our 2,3-BPG, it's made from an offshoot of glycolysis. it binds to the central channel that's only available in the T state, so the non-oxygen bound form, as opposed to the R state, the relaxed form, the form that is actually going to be bound to oxygen. So binding of the 2,3-BPG to that central channel is going to make it so that the hemoglobin is going to be stabilized in that T state, that oxygen offloaded state, and it's going to be harder for it to pick up the oxygen. Whether or not it picks up oxygen is then going to kind of depend on how much oxygen is around as well, which is why you can still have even if you had like similar levels of 2,3-BPG, you could have the amount of oxygen that's actually present is going to make a difference. 2,3-BPG is binding in that central channel between the beta subunits, or in fetal hemoglobin, those would be your gamma subunits. And in the gamma subunits, you don't have as many favorable salt bridges that can form, and so fetal hemoglobin is going to have a lower affinity for that 2,3-BPG. As a result, it's going to be less affected by the 2,3-BPG. And therefore, you're going to basically get a left shift if you were to compare it to the adult. But really, that kind of like left shift is just an artifact of it just being less of a shift to the right, because 2,3-BPG would shift it to the right. And so you're not shifting to the right as much. And so you're going to be have a higher affinity. In the presence of BPG, you'd have a higher affinity of the fetal one than the adult one, which is good because what's going to happen is that the fetus is going to be exposed to a lower concentration of oxygen than the adult hemoglobin is. Another key thing about 2,3-BPG is that its production is upregulated, so there's more of it made at high altitudes. And this is important because there is less oxygen available at high altitudes. So it might seem a little counterintuitive that you're going to kind of decrease your affinity for oxygen when there's in places where there isn't very much oxygen. But that's because you have to kind of think about the shape of the hemoglobin curve. Typically with hemoglobin curves, you're often comparing two places. And so you're comparing like, okay, well, I've got the oxygen in my lungs versus that in my tissues. And that's going to tell me how much am I actually releasing. If we were to look at the amount that we would be releasing, if we only had a low concentration of BPG, well, what would happen is that in our lungs, we'd be starting with less, but then we'd be getting to the same point in our tissues. And so we'd be releasing less. We'd be releasing less than if we had started with the pressure of oxygen, so the concentration of oxygen that you see at sea level. So there you would get 38% versus here you would only get 30%. But if you increase the BPG, well, now you're going to shift things right. And if you shift things right, yeah, you're going to decrease. So you're picking up a little less in your lungs. You're a little less saturated in your lungs. But then because the curve is still kind of like flattish up here, but then when you get to this kind of big drop, well, now you're shifting the position of the big drop. And so you end up having a big change, a big difference between the different concentrations of BPG at this lower concentration of oxygen, at the concentration of oxygen that you find in the tissues. So the concentration of oxygen that's found in the tissues isn't changing. It's the concentration that the lungs are exposed to that's changing when you think about sea level versus high altitude. So at high altitude, you're going to have less available in your lungs. So you're starting with less. You're starting with even less of that is going to actually be bound to hemoglobin if you have the 2,3-BPG, if you have more of the 2,3-BPG. But that's a small kind of decrease compared to the decrease that you're going to see in your tissues, the decrease of affinity, that is not the decrease in like the oxygen concentrations. You're getting to the same oxygen concentration, but now you have a lower affinity. And so you're going to be offloading more. And so in this way, the BPG is able to kind of compensate. So you have a little less here, but you have a lot more here. And so you're going to overall be releasing a lot more oxygen, delivering a lot more oxygen than if you had this condition where you were starting here, but without as much BPG. And in that case, you're only getting this 30% versus the 37% you get with the higher concentration of BPG. People in a high altitude are going to have more 2,3-BPG because basically it's going to increase the amount that gets offloaded to the relatively same amount as you would normally.